Good morning everyone. This is Jyoti Pandey and today's lecture topic is description of phase contrast microscope. This topic includes the description of phase contrast microscope, their importance, their types and their applications. This is the diagram which represent the phase contrast microscope structure. So what is microscope? Microscope is an instrument that is used to see the object which are very smaller in size and cannot be seen via the naked eyes. In the science, to see the smaller objects, there is a device which is known as microscope, and the science is known as microscopy. Types of microscope. There are variety of microscopes present in the field of science. First one is optical microscope. This type of microscope uses the light to visualize the image of any object. Second one is electron microscope. Such kind of microscope uses the electrons to visualize the image of any kind of sample or object. Electronic microscope includes certain subcategory of a microscope like scanning electron microscope, transmission electron microscope, ultra microscope and scanning probe microscope. What is light microscope? Light microscope are the category of microscope which are uses the visible lights to detect the smaller objects. It is also useful in the field of biology to magnify to magnify and to visualize the cellular part of any kind of smaller organism, protozoans, unicellular organisms, etc. like bacteria. This is a light microscope which is useful to enlarge cytoplasm, nucleus, cell membrane, chloroplast, vacuole, cell wall, etc. components of any multicellular cell. Now comes to magnification. Well, magnification and resolution, these are the property of microscope. Magnification means degree of enlargement. This decreases the number of times in length, breadth and diameter of any object as multiplies. Resolution, what is resolution? Resolution is the minimum distance between two visual bodies at which they can be seen as separate and distinct objects and not in contact with each other. Types of light microscope. Light microscope is characterized into a three category. First one is bright field microscope, second is dark field microscope and third one is phase contrast microscope. Bright field microscope produces a dark image against a brighter background. Dark field microscope produces a bright image against a dark background. Phase contrast microscope is important microscope which is comes under your syllabus profile of microbiology. So phase contrast microscope is used for enhances the contrast between the intracellular structure having slight difference in refractive index. So intracellular structures को और अच्छे से विजुलाइज करने के लिए फेस कॉन्ट्रास्ट माइक्रोस्कोप को जनरली यूज किया जाता है. It enhances the contrast power. So there is a comparison between the diagram which was provided by the light microscope. This is the bright field image, this is dark field image and this is phase contrast image which is obtained after the microscopy from the different kind of light microscope. Now here is a description about the phase contrast microscope. This is a microscope which is useful for determination of their microbial structure under the contrast media. This methodology first of all observed in the living cells by the scientist Fritz Czerny in the year of 1953. and he was awarded with a Nobel Prize in 1953 for his work. Phase contrast microscopy. This is a microscopy made it possible to study the living cells and how they proliferate through cell division. So the cell division can also be studied with the help of phase contrast microscopy. It is also used to examine living organisms or specimens that would be damaged or altered by attaching them to slide or staining. It also uses the conventional light microscope fitted with a phase contrast objective to see the phase contrast images. Light passing through one material and into another material of slightly different refractive index or thickness will undergoes or visualize in phase contrast microscope. This change in the translated and variation of structure is easily differentiated with the help of phase contrast microscope. This is the flow diagram of phase contrast microscope. This is a phase plate. This this is diffractive light segment. This is specimen. This is unobstructed. This one is unobstructed light, or we can say phase unaltered by the specimens. What are the principles of phase contrast microscopy? It involves diffraction of light. Unstained bacteria have constitute of different refractive index. Phase contrast microscope employs an optical mechanism to translate minute variation in phase into corresponding changes in intensity of image. So as per the intensity of image, this type of microscope translates their variation into a phases. These are the phases: race in phase, race out of phase, and phase plate. This one is phase plate, which shows the phases in a phase contrast microscope. This is a lens, this is a phase plate, and this is a sample, object sample. When passes the rays, then ray deviated by the specimen is about 1/4 wavelength out of phase. Then it passes to the phase plate, and deviated ray is now 1/2 wavelength out of the phase, and the image was shown in lens. What are the uses of phase contrast microscopy? This is useful uh, in the visualization of internal cellular component. It is also useful for the diagnosis of tumor cells, for the examination of growth, dynamics, and behavior of a wide variety of living cells in cell culture. Other application includes phase contrast microscopic equipment range from the study of living biological specimens, medical application, study of live blood cells, and other biological and science applications. Most commonly, it is used to provide the contrast of transparent specimens such as living cell or smaller organism. There are some references which useful for the further study and for the detailed study about the phase contrast microscope and types of different microscopes.